Hello guys, and in this video we'll be discussing the IP3 signaling pathway. We're going to talk about how protein kinase C gets activated. And as you already know, a kinase adds a phosphate. So we'll have our G protein here, seven transmembrane domains. You can count them 1, 2, 3, 4, 5, 6, 7. That's our G protein coupled receptor, and our G protein is actually located intracellularly. And I drew GTP here to indicate that that's what is bound whenever the G protein is active. So our ligand here, let's say, binds to this receptor, right? It causes a conformational change, which eventually leads to the alpha subunit acquiring GTP and becoming active. Once this alpha subunit is active, it's going to dissociate from the rest of the complex, the gamma and beta subunit. It's going to activate something called phospholipase C. This is phospholipase C right here. I'm going to put some whatever they do this to indicate something is active, like whatever, I don't know how to describe it, but yeah. So the phospholipase C becomes active, and once it's active, it's going to cleave this membrane-bound phospholipid known as PIP2 into IP3 and DAG. DAG will remain membrane-bound and serve as a docking site for something called protein kinase C. And that's basically this video where we're talking about how it becomes activated, right? So it's a docking site. That means that DAG will come here and basically dock. And then let's talk about the rest of the story here. So IP3 is going to go to not sarcoplasmic, well, that's for the muscle cell, but endoplasmic reticulum, and is going to cause the release of calcium. Calcium 2+. Right? And so what calcium is going to do is it's going to go here and bind to PKC and cause it to become fully activated. Well, it's not active then, but just it becomes activated. I'm saying fully because first it had to dock, and now it's bound to calcium. Now it's active. So it's a kinase. So what do you think is going to happen to it now? It's going to be able to phosphorylate target proteins and cause a change. So I'm going to put a P here for a phosphate. PKC can now phosphorylate target proteins and cause some kind of change inside the cell. So let's go back from the beginning and work through this again. So you have your ligand that's going to bind, is going to activate a G protein. This G protein is actually, if you want to be technical, it's a GSQ. That's going to activate phospholipase C once the alpha subunit dissociates. Phospholipase C is going to cut a membrane known as PIP2 into IP3 and DAG. DAG will serve as a docking site for protein kinase C. IP3 will then go to the sarcoplasmic reticulum, bind, and cause the release of endoplasmic reticulum. I keep going with muscle cell. I apologize. So the sarcoplasmic reticulum will release calcium. Now calcium can go and bind to protein kinase C and cause it to become fully activated. And as a kinase, what it does, a kinase adds a phosphate. So it's going to phosphorylate target proteins and cause cellular change. Thank you for watching, and if you have any questions, please send them in, and don't forget to please subscribe.